Good afternoon, and thank you for joining us for the very first of the research webinars from HDSA for 2021. Today, we're delighted to hear from two very familiar faces, Dr. Sandra Kostyk and Dr. Andy Feigin. They're going to talk to us about the PROOF-HD study. But before I give them a formal introduction, I just wanted to remind everyone that you can ask questions at any point during this webinar using the chat function. So just down in the bottom of your screen, click chat, ask your question, type in your question. At the end of Dr. Feigin and Kostyk's presentation, Dr. Leora Fox, my colleague who's also joining me, she and I will be asking the questions on your behalf. This video and webinar is being recorded. So within about a week's time, if you have to tune off or you have some loved ones that you think could benefit from hearing this, you can go to our YouTube channel, which can be found at the top of our hdsa.org page, and all of the research webinars, including this one and many others, can be found there. Just take a minute to announce that next month we'll have another research webinar, and it's going to be myself and Dr. Leora Fox. We're going to give a 2020 HD research year in review. And I believe in March, we've got one planned, and we'll get more updates in February, but we've got one planned on another clinical study, the Connect HD study. Is that right? It's Connect, right?
Yep,
Leora?
more details to follow.
Great. All right. So that's all for an announcement. So it's really my pleasure to introduce our two speakers today. First, Dr. Sandra Kostyk. Dr. Kostyk completed her MD and PhD at the University of Chicago in her neurology residency at MGH. She's currently the medical director of the HDSA Center of Excellence at Ohio State, and has been there since 2000. She's participated in numerous Huntington Study Group, industry, foundation, investigator-initiated HD trials, and she's dedicated to increasing HD awareness and education and improving care options for individuals and families affected by HD. And our other speaker is Dr. Andy Feigin. Dr. Feigin's been involved with HD clinical research for more than a quarter of a century. 25 years is a long time. He's participated in the Venezuela HD project for 10 years, beginning in 1993, the year that the Huntington's gene was identified. And he's been a site principal investigator for more than 30 NIH and industry-sponsored clinical trials for new treatments for both HD and Parkinson's disease. In addition, Dr. Feigin's a principal investigator for several multi-center clinical trials for HD and PD. He served in several leadership roles for the Huntington Study Group and currently serves as the chair of that organization. In addition to his clinical research activities, Dr. Feigin maintains a busy clinical practice caring for patients with HD, Parkinson's, and other movement disorders. He's currently a professor in the Department of Neurology at NYU. So with that, I'll turn it over to, I will stop sharing, and I'll turn it over to Andy. You can share your screens and get us started.
Okay, well, yeah, thank you so much, George, and thank you to the HDSA for inviting us to talk about the PROOF-HD trial. So I'll turn it over to Sandra to start.
Andy, you're going to have to get up onto the first slide. And
oh, okay, sorry about that. Let me do that. Yep.
The slideshow.
Here we go.
There we go. So we welcome you all today, and we appreciate this opportunity to share information about the PROOF-HD trial and our enthusiasm for why we're proceeding with this. The trial is sponsored by Prolenia, and it's being run in conjunction with the Huntington Study Group, and it is an international trial, so will be sites in the United States and in Europe. The title of the trial is officially Pridopidine Outcome on Function in Huntington's Disease, and we shorten the name to refer to this trial as PROOF-HD. I'm going to read you the next sentence just to give you a sense of how we name a trial. So this is a phase 3 trial, which implies that we've already successfully gone through phase 1 and phase 2 of moving this agent to FDA approval. It's randomized, double-blind, which basically means it's placebo-controlled, and their two arms are equal. It's a multi-center study, and our goals are to evaluate efficacy, which means the effect of the agent, and safety, and the agent is Pridopidine in individuals with early stage Huntington's disease. We're going to go through the rationale for why this is an agent of such interest and try and explain the study to you a little bit. Dr. Feigin and I are going to kind of alternate slides, and I'm going to turn the next slide over to Dr. Feigin.
Thank you, Sandra. Yeah. So first of all, thank you to all of you for joining today and for your attention. And once again, just want to thank the HDSA for allowing us giving us this platform to come and talk about this trial. So I thought we would start just talking about the study drug Pridopidine and why we think it might be helpful for people with Huntington's disease, what its mechanism of action is. First thing I would like to say about Pridopidine is that it has been the subject of actually three large prior clinical trials, one called Heart, one called Mermaid, and one called Pride. Those trials were done at a time when the mechanism of action of Pridopidine was thought to be different from what it turns out the primary mechanism of action of the drug is. So those trials were actually aimed at identifying an effect of the drug that is different than what we're going to be talking about today. Since that time, it's been discovered that Pridopidine works on something called the sigma-1 receptor in the brain. And in fact, it works almost exclusively on the sigma-1 receptor. It's very, very specific to the sigma-1 receptor and binds much less to other receptors in the brain. And there are, as we'll show you, there are many reasons to think that drugs that act through the sigma-1 receptor should be helpful to patients with Huntington's disease.
So the sigma-1 receptor looks like it has multiple effects on cells in the brain. HD is a complicated disorder, and we still do not actually understand all the mechanisms of action and how it is that the mutant Huntington protein disrupts cellular function. But we do know a number of features of cellular function are significantly impaired in Huntington's disease. What we've learned now, though, is that the activation of the sigma-1 receptor has multiple mechanisms of action that should lead to neuroprotection in HD and help stabilize cell function. We know that calcium function and homeostasis is dysregulated in HD, as sigma-1 receptor activation can mitigate that. It affects mitochondrial function. It looks like it can improve neuronal plasticity, which means how the nervous system responds to both the internal environment and things going on in the external environment. There's evidence to suggest that it increases BDNF levels. BDNF stands for brain-derived neurotrophic growth factor. And there's evidence in HD and in other neurodegenerative diseases that this agent, the small neurotrophic growth factor, is critical to cell survival and responding to negative impacts of things like the Huntington protein. Activation of the receptor decreases oxidative stress, improves dendritic spine, or increases dendritic spines, which reflects a way of how cells interact with each other in the nervous system. So there's multiple positive effects of this activating this receptor in the brain. The other reason we think that there's that just sort of reinforces our enthusiasm for why this is a good agent to be looking at. On the right side of the screen, there's sort of a picture of the sort of a modeling system of the distribution of the message that makes this receptor in the brain. And in HD, the first part of the brain that is most seriously affected by the Huntington's disease mutant protein is in the striatum, which is composed of the caudate and the putamen. So looking at this kind of image, what we see is that in the caudate and putamen, colors that are red and orange are where the receptor would be in high concentration. In the striatum, there are numerous sigma-1 receptors, and not only is it in the center of the brain in the striatum, but the whole cortex lights up highly with signs that the sigma receptor is present and can be activated throughout the cortex. Next slide.
So one of the ways that we sometimes try to show if a drug might be helpful to patients with Huntington's disease is to study it in animal models of Huntington's disease, and specifically in what we call transgenic animal models of Huntington's disease, animals that have been bred to have the Huntington mutation in them. And so on the left panel of this slide, what we're showing is in blue, mice that do not have the Huntington mutation, and in orange, animals that do have the Huntington mutation. And what you can see is that in the blue bars show that there's a relatively low level of cell death over time in animals that don't have the Huntington mutation. But in mice that do have the Huntington mutation, if you look at the column that says 0, that's 0, they have not received any of the study drug, Pridopidine. You see that there's a lot more brain cells dying in those mice, the mice that carry the Huntington gene mutation but have not been treated with the study drug. And then if you move along on this, see if I can point this out. So if you move along this way in this graph, you go with increasing concentrations of the study drug, Pridopidine. So as you go to the right, you end up with a higher concentration of Pridopidine. And what happens is the more of this drug that you give to these mice that carry the Huntington mutation, the more that these mice start to look like mice that don't have the Huntington mutation in terms of the brain cells that are dying. That is, that the Pridopidine seems to be protecting brain cells from dying, and in a dose, what we call a dose-dependent way. The higher the dose of the drug, the more that these animals, the brain cells that are dying starts to look just like the way the brain cells die in mice that don't have the Huntington mutation. On the right panel, what this shows is that if you create mice that don't have the sigma-1 receptor, that is, this is called sigma-1 receptor knockout mice. So these are mice that are bred to not have the sigma-1 receptor. And you do the same experiment that you did over here. So except now the mice don't have the sigma-1 receptor. Still, the blue mice don't have the Huntington mutation. The orange mice do have the Huntington mutation. And so they're losing more brain cells than the blue mice. But what happens is when you go up on the dose of Pridopidine, you don't get any protection. And that's because these mice don't have the sigma-1 receptor. So it shows that this protective effect of Pridopidine is mediated through binding to the sigma-1 receptor. I hope that's clear.
So being convinced that this has potential, we also have a lot of confidence that this is a safe and a good medication to start using in Huntington's disease. Because it's been studied in over 1,300 subjects, most of whom had Huntington's disease, we have a lot of data that helped us choose the dose to give for the study and also to feel comfortable that this is a safe and tolerable medication. So it's been studied in humans from low doses of 10 milligrams twice a day to high doses up to 112 milligrams twice a day. It's across the range, it proved to be safe and tolerable based on some additional analysis of outcomes and functions. The dose of 45 milligrams twice a day was chosen for this study. And that dose appears to be quite safe. There have been, because it's been used in multiple trials, and Dr. Feigin will talk about one of these in particular in the next few slides, we're comfortable that this is a safe agent for individuals with HD. In the prior studies, when the placebo, the pill that did not contain Pridopidine, was compared to the complaints that that people have over the course of the study, there were no significant differences. There would have been about the same amount of people reporting that maybe they thought the drug caused headache or stomach ache or fatigue. There was nothing that stood out as a particular side effect related to this agent. Next slide, Dr. Feigin.
Yeah. So we're going to shift gears a little bit to talk about some of the clinical data that we have seen in prior trials with Pridopidine that kind of backs up what we've already showed you and suggests that maybe this drug really could have a positive impact for patients with Huntington's disease. And to do that, I thought it's important to talk about what we call the primary outcome measure of our trial. That is, when you do a clinical trial to show if there's an effective drug, you need to, especially a phase 3 trial, you need to pick one outcome measure that you say is the most important outcome measure, and you're going to hang your hat on the result with that outcome measure. And in this case, we've decided to use an outcome measure called the total functional capacity, or TFC. The total functional capacity is a measure that's actually been around about 40 years. It was developed, I think, almost 40 years ago. It's been used in numerous trials, both observational trials in Huntington's disease and also in clinical trials of Huntington's disease. And it's been being used in a lot of current clinical trials, including trials that you have probably heard about, like the Roche ASO trial. The TFC is a major outcome measure in that trial as well. This measure, the total functional capacity, is part of the Unified Huntington's Disease Rating Scale, and it measures the burden of Huntington's disease on people's functioning, how well they can stay at work and perform at work, how well they can manage their finances, how well they can manage their chores at home, or can they be taken care of at home. All these kinds of measures of how people function in their daily lives, things that are meaningful to patients with Huntington's disease. And the FDA has recognized that this scale is important as an outcome measure by stating that what is stated here: the UHDRS total functional capacity is the most clinically relevant scale to measure maintenance of functional capacity that has been determined to have the most impact on patients' daily lives. And I just wanted to emphasize this because this is the primary outcome measure for the PROOF-HD trial. And we've looked at how the TFC has responded to Pridopidine in past trials. And so we want to kind of go through that with you as further evidence that this drug, Pridopidine, may be helpful.
So when we look at the TFC, which we're saying is a global assessment of how people are doing, living at home, how much help they need, what they can do, we have years of data. And as Dr. Feigin mentioned, the scale was designed probably 30 to 40 years ago. And we've looked at it and watched in observational studies to see how the score changes over time in thousands of patients. So this is sort of a summary of what we look at. And what we've learned is that on average, in individuals with earlier stage disease, the total TFC score goes down, total functional capacity score, by about 1 point per year. So both in the very early and the mid-early stage, the average score decreases by 1 point. Later stages, the TFC score may not be as reflective of the rate of decline, and the decline is recorded as less. So to help facilitate getting this trial validated, there are some restrictions to who would be enrolled in the trial, and the individuals with earlier stage disease will be participating. This does not necessarily mean that Pridopidine may not be helpful in later stages, but in later stages, using this particular outcome measure, it would take much longer to prove effectiveness. And our overall goal is to get this agent FDA approved as soon as possible so that we can start using it in individuals, if it's proven beneficial in individuals affected by Huntington's disease.
Yeah, you know, Sandra, I should have mentioned, and maybe I'll mention on this slide, that the scale goes from 0 to 13, which is kind of suggested here. 13 is the highest score you can get on this total functional capacity. And essentially that means that a person is working at a normal capacity and doing all the other activities that are measured in this scale normally. And then, as you mentioned, in people with mild, relatively early Huntington's disease, the scale declines on average by a rate of about 1 point per year. So in one of the prior trials that I mentioned of Pridopidine in Huntington's disease, that trial was actually designed to look at a different aspect of Huntington's disease and not at TFC. TFC was measured though. And so when we look back at how TFC changed in that prior trial, what we found is that if you were in the placebo in that trial, Just as predicted from other observational studies, the placebo patients declined at just around 1, maybe a little bit more than 1 point per year. If you happen to be randomized to 45 milligrams twice a day of Pridopidine over the course of a year, there was essentially, on average, essentially no change in the total functional capacity. And this was highly statistically significant, this difference. So I think it is important to mention here, first of all, this looks very promising and very interesting, and is one of the major reasons we think that this drug may be helpful for people with Huntington's disease. But somebody who's not familiar with clinical trials might look at this and sort of say, well, here you did it. You showed that the drug works better than placebo, and why are you doing another trial? And that kind of goes back to something I mentioned earlier, which is that trials need to be designed to look at specific things. And the PRIDE trial was actually designed to look at a different measure, to look at motor function actually in Huntington's disease, not at functional capacity. And so even though there was this observation, this was done in what we call a post hoc way and couldn't be used for getting approval of the drug. But it is helpful in terms of getting a sense for that the drug may have had an effect in patients with, again, this is with patients with a total functional capacity of greater than or equal to 7 who participated in the PRIDE trial.
So just to sort of define a little bit more about the total functional capacity and the effect that when, again, Dr. Feigin mentioned this concept of post hoc analysis. And this is when we're trying to get a drug approved, the FDA has certain hoops you have to run through, and you choose a primary outcome measure. And of the other trials, they were looking at other measures of the disease. But after the trial was completed, we were able, I say we as in the whole global HD research community, to look at the data from the original trials and tease out parts of that study and get information that sort of gives us an idea of how to look for a better outcome measure. And as he said, and as we've going through, total functional capacity is a measure that's really important to people's lives. From the voice of the patients, it tells you a little bit about the things you're concerned about. When can I still work? Can I still drive? Can you do things? It does have 5 components. And what's promising is that each of these components was to some degree affected by beneficially in the previous trials. Ability to do work, household activities, activities of daily living, which might include things like showering, bathing, preparing meals, finances, balancing checkbooks, ability to go to the store and buy things independently, and care level, how much assistance you need at home or in a facility. So all of those components were impacted to some degree in the preliminary trials and the phase 2 trials that were completed to date.
And just to kind of give you a sense for how the TFC has fared in other trials that have been done over the years. This is just a sample of some prior clinical trials. There have been many, many clinical trials in Huntington's disease, but just to give you a sense of what's been observed in these trials, both in placebo groups and in the treatment groups. We've listed some observational trials, at the top here. That the Huntington Study Group has done. If you look at the column called annual TFC progression, that's really the most important column here. And if you just kind of look down here, you see in these observational trials that the TFC declines anywhere from around 3/4 of a point to 1 point to just under 1 point, anywhere from 0.7 points per year to 1 point. Actually, in the PRIDE trial, if you were on placebo, is maybe around 1 point or a little bit more than 1 point that it declined over the course of a year. In CARE, HD was a trial that stood for coenzyme Q and racemide efficacy in Huntington's disease. This was a trial that looked at high doses of coenzyme Q10 and a drug called racemide to see if they might affect the progression of Huntington's disease. And coenzyme Q, people who were taking coenzyme Q progressed at about 1 point a year. People who were taking racemide progressed at about a point per year in the TFC. People in a clinical trial of creatine in Huntington's disease, people progressed at a rate of about 0.8 points per year. People in the tetrabenazine trial progressed at a rate of about 1.3 points per year on the total functional capacity. So very consistent progression in the range of 0.75 to 1 point on the TFC per year. The one standout observation was what we already showed you, which is that if you were in the PRIDE trial, the trial of Pridopidine, and you had a TFC to start with between 7 and 13, and you were taking Pridopidine 45 milligrams twice a day, over the course of a year, your total functional capacity really didn't change. So it's a remarkable difference from all these other observational trials, both in and treatment trials, both in the placebo and the treatment groups. And this has led us to think, well, maybe this bit and the data from the animal model of Huntington's disease that we showed you has led us to think that this is something worth revisiting, to try to see if people who take 45 milligrams twice a day of Pridopidine can maintain their functioning better than people who are randomized to placebo.
So another thing that's, I think, important to us when we think about something that we call a disease-modifying intervention, something that slows the course of the disease, you're going to be anticipating that this is not something you're going to be giving someone for a short period of time, just for 12 months, but perhaps unless there as other therapies, treatments, or cures come on, people will be on this agent, if it's proven beneficial, for years. So fortunately, again, because of the way this drug has been developed and studied, we do have some data over time, over 5 years of time, and we can compare that to the placebo used in other trials. So the 2CARE trial was actually a 5-year-long trial. And as you look at the last column after 60 months, the scores, as expected, and this also just shows how robust this tool is as a measure. The scores went down by about 5 points over the course of 5 years. Now, the number of individuals in the open heart Pridopidine trial was not as great, but nonetheless, looking at 55 individuals, if you go again to the 60-month trial, there was a significant decrease in the loss of scores on the TFC compared to placebo. So this is not just a short-term effect. This supports the possibility that this agent will have long-term beneficial effects over the course of as many as 5 years or more for individuals affected by Huntington's disease.
So just to give you a few details about the PROOF-HD trial, which is now underway, and patients are being enrolled, screened and enrolled in the trial as we speak. As Sandra discussed at the beginning, this is a phase 3 parallel arm randomized double-blind placebo-controlled multi-center study evaluating the efficacy and safety of Pridopidine in patients with early-stage Huntington's disease. There are two arms in the study, placebo and Pridopidine 45 milligrams twice a day, and they're equal, so that this slide doesn't say it, but 480 patients will be enrolled in the trial. Half will get placebo and roughly half will get Pridopidine 45 milligrams twice a day. Because of the data we showed you, we're limiting the trial to people with relatively early to moderate Huntington's disease. That is total functional capacities at the beginning of the trial of greater than or equal to 7. The primary endpoint, as we've already discussed, will be the change from baseline in total functional capacity to week 60, actually I can't see that, is it 65 or 65? Yeah, 65, which is a little over a year. And then we'll be doing a bunch of other types of analysis that involve total functional capacity. For example, we'll be looking at something called a responder analysis. How many patients in the trial over 65 weeks showed no change, no worsening in total functional capacity if you're on the drug versus on placebo? Is there a difference between those two measures? And we'll be looking at other endpoints that measure the progression of Huntington's disease as well, in terms of motor and cognitive measures and other quality of life measures. Importantly, I think we've also included in the study things to mitigate for potential complications related to COVID-19. That is, If there continues to be problems with COVID-19 and study sites need to shut down, we've actually included the possibility in this trial that some of the visits can be converted to telephone visits, where we'll do the total functional capacity by telephone. We've also actually made arrangements, if we need to, for some of these studies visits. If people can't come to a study site, of course, we want people to come to the study site for all of the scheduled visits, but if they can't for one reason or another, we've also made arrangements for safety measures like EKG and blood tests to be done at people's homes. So we're pushing ahead with this trial, even under these difficult circumstances, because we think it's an important trial and we don't want to wait to get started. And we've made some of these mitigating arrangements in case we need to fall back on not having people come for every visit to the trial sites. One thing I want to mention on this slide, which is also not on here, so I mentioned 480 patients. It will be at 60 sites, 30 in North America, US and Canada, and 30 in Europe as well.
There's also another thing that we didn't mention on that slide. Actually, if you want to just go back,
oh, yeah, sure.
We can talk. The visits are kind of spaced out. So over the course of the year, there are probably about 11 or 12 visits. And in terms of the mitigation, we hope that there are sort of about 4 visits that are absolutely essential, but we are making accommodations, as he mentioned, as Dr. Feigin mentioned, in case there are problems, and we cannot predict anymore these days how the world is running. But the other thing which we did not mention here, and Dr. Feigin and I forgot to sort of talk about as we were putting our slides together, there is, after the trial, a open label extension, where everybody in the trial will be able, whether they're on placebo or study drug, keep on the study drug or switch over to study drug at the end after the 65 weeks, a little washout period, and people will have the opportunity to stay on the study drug until it's FDA approved.
Yeah, thank you for mentioning that, Sandra. That's an important point. Yeah, thanks.
And I think our last slide is just to point out that this is actually a big collaborative effort. It's an international effort between the Huntington Study Group, the Pronilia, and it's required a lot of work together. And in the North American study, Dr. Feigin is the primary investigator, and I am the co-investigator. In Europe, Dr. Ralph Raumann, who is the founding director and CEO of the George Huntington Institute in Munster, is the primary supervisor of the study in Europe. And his co-PI is Dr. Ann Roser, who is the chair of the European Huntington's Disease Network and a professor of neurology at Cardiff University. So moving this forward is a big international effort, a team effort, and we look forward to seeing the study advance. There, at the bottom of this slide, but there are many ways you can access this. You can find out more about the study, where the sites are, by going to the website listed here. And for those of you who just rather talk to a human being, there is a 1-800 number dedicated to the PROOF-HD trial. And thank you very much, and we're happy to answer questions.
Yeah, thanks so much for your attention. Really appreciate it. And as Sandra mentioned, yeah, we're happy to take some questions.
Thank you, Sandra and Andy. We've got a lot of questions coming in, so please do keep them coming. And thank you for sharing how to get involved. There are a number of questions that I'm seeing about how to get involved, who do they talk to, and certainly the Huntington Study Group website and phone number is one way. We also have this site listed in hdtrialfinder.org, which will list the currently recruiting sites for the PROOF-HD study in North America. And there's also a site, I know there's some folks calling in and dialing in from Europe today. So unfortunately, HD Trial Finder does not list sites in Europe, but there is another HD Trial Finder website in development that should be up and running that may have some of this information for some of the European sites. And in addition, all of these sites are listed and posted in clinicaltrials.gov. where you can get a lot of the information that Andy and Sandra just presented.
So I'm getting a sense there's a fairly enthusiastic response to possibly participating in the study. We're talking to you very early in the study, so not all sites are up yet. We're just starting recruitment, and some of the sites may not yet be listed actually on clinicaltrials.gov. I don't think that they're all there yet, but if you keep in contact, especially through the Huntington Study Group in the United States, you should have an idea of who's started up already and who's in the process in the near future.
Yeah, as Sandra mentioned, we're in the process of getting the sites up and running, and we're expecting really in the next month or two to have a real kind of big step up in the number of sites that are active. Yeah.
I can also confirm that we have been working with HSG to list the trial information in hdtrialfinder.org for all the sites that are currently recruiting. We're getting a lot of questions that are related to eligibility. Tons of great questions here. And one main question has been, can folks be on other drugs while participating in this study? For example, drugs for chorea or for depression or anxiety?
So the answer is that many of the drugs are allowed. There are some drugs that are not allowed, and there's a list of inclusion and exclusion criteria. Rather than going through that in detail, which partly reason I don't want to do it is I don't have it in front of me. Many drugs are allowed. Drugs that are commonly used in Huntington's disease are allowed. For example, tetrabenazine. I don't know if people are wondering about that. Tetrabenazine is allowed,
almost all of the antidepressants and the behavioral medications, not literally everyone, but most of them are actually allowed.
Yeah. So we're eager, it's a big task to enroll 480 patients in a clinical trial. So we designed the trial to be to cast a broad net, and that doesn't mean everything is allowed, and so there are some exclusionary medications, but the best way to kind of approach that, I would say, is if you're interested, is to find a site near you and to have a discussion with the staff at that site.
Questions coming in related to the primary endpoint of the study, which is TFC, and they're inquiring about does that score change depending on the doctor who does the actual analysis? So maybe one of you could address that.
Yeah, it's a really good question. And it's an issue that comes up with all of these clinical scales that we do. And so what we do is we do training and credentialing for people who do this scale to make an effort to make sure that everybody is being consistent in terms of with each other, but also with themselves, actually. So we have extensive training annually, and also specifically targeted at this trial, where we go through how we would approach doing the total functional capacity, what would constitute a score of different parts of it. And also, I think a related question that we've also discussed at length is when we do the TFC, do we do it with a family member or caregiver as well as the patient, or do it with the patient alone? And so we try to be as consistent about that as possible. If we're doing it with a caregiver, then we want it being done with a caregiver at every visit. If it's being done by the patient alone, which is allowed, we want it being done by the patient alone at every visit, to get at this issue of being as consistent as possible.
And we also really tell the sites strongly prefer that the exact same evaluator does this particular score. Right. And even the other scores, the cognitive testing, have a dedicated person for doing that. Many sites have several investigators, but we prefer and we strongly instruct actually at our investigator coordinator meetings to have the same person administer each portion of the test at each and every visit.
Thank you.
Thanks. Several people have asked how this drug is administered. Is it a shot? Is it a pill? Can they take it at home? How does that work?
It's a pill, and you can take it at home. And in fact, actually, you know, when I mentioned the COVID mitigation strategies, I talked about safety measures potentially if people can't get to the office because the offices are closed. I mentioned that, you know, you could do EKGs and blood work, but we're also making arrangements, if that were to happen, of actually being able to send the medication to people's homes, actually.
So it's one pill twice a day.
Yeah, one pill twice a day, by mouth, yeah.
Very easy.
Yeah.
By mouth.
Yeah.
That you take it home.
Few questions also coming up, inquiring about maybe some side effects or adverse events that were noted in previous studies that tested Pridopidine. Can you address any of those adverse events that may have emerged in previous studies?
There were actually very few, and whenever you run a clinical trial, you'll have people have the report. You're supposed to report any medical complaint you have. And for instance, I did a trial and someone stubbed their toe and lost their toenail. Now, was that related to the study drug? Probably not, but you record it as an event. People will have headaches. That happens, or they'll have gastroenteritis or diarrhea or viral flu. In this data from the multiple studies that have been done looking at Pridopidine, there was no significant outstanding complaint in the placebo group compared to the study group. So there's not something that we are expecting to be very different. And side effect profile of this particular agent is incredibly good. And not only is the side effect profile good, if you look at some of the other data that we didn't show you here, there have been studies looking at compliance and how many people drop out of the study. And as I recall, the table that I looked at a while ago, of 8 trials in Huntington's disease, you're going to expect some people to not be able to continue the trials. But the Pridopidine trials had the best retention of all the studies that have been done, pretty much the best of all the studies that have been done so far, which also suggests that there weren't a lot of side effects making people want to stop the study drug during the course of the trial.
Yeah, I'll just add that, I mean, as Sandra alluded to, when we do these trials, we don't usually speak about side effects. We speak about adverse events.
Mm-hmm.
And so since we're blinded to whether people are on the drug or on the placebo, we just report adverse events that occur. And then at the end of the trial, look to see if they're different between the placebo and the, but it's really not possible to make a judgment about cause and effect. It's really a matter of what events occur in one group versus what events occur in the other group. And we do have a safety monitoring committee that is able to look at the distribution of these events as they occur in real time during the conduct of the trial. So there is a kind of a layer of people looking at this as it's going on. So if something were to turn up that we didn't expect, we would find it.
Thanks. We've had several questions relating to when this drug might be available to folks if it were to be successful. And I know that's a very tough question, but maybe you can speak a little bit to the timeline of expected full recruitment and when you might expect results from this study.
Well, you know, when we set out to do a trial like this, we always want to recruit as fast as possible. You know, I mentioned already that it's 480 patients at 60 sites. So if all the sites get up and running and recruit, each site on average would be expected to enroll 8 patients, which doesn't sound like so much. So our goal is to complete recruitment by the end of this calendar year, actually. You know, so we'll see whether we can achieve that, but that is the goal. And then the trial lasts 65. The primary outcome measure of the trial is at 65 weeks, so that's another year and a quarter roughly. And then, you know, so that part of it is, you know, not perfectly predictable, but at least we have an idea about what our goals are and what we're aiming for. The hard part about it is, you know, after that, we analyze the data, submit, you know, depending on whether we hit the primary outcome measure or not. If we do, then it has to go to the FDA where it gets reviewed, and that the timeline for that can be quite variable. So it is a little hard to predict. But I would say, you know, from today to the completion of the trial, we're talking about probably, between 2 and 2.5 years, and then sometime after that, having the data analyzed and proceeding to the FDA if it works.
So some questions about age. That's important, and it's been an issue for folks in other previous HD studies. But there's some questions coming in about, can my child who has JHD, who's 18, participate, as well as the upper limit? Can you address the age qualifications for this study?
Yeah, the inclusion criteria for the trial are you have to be, I think it's 25 and up, actually, is the age. And part of the reason for that is, I know that that we all feel like we want to do trials that are going to be inclusive of people with juvenile Huntington's disease. It's a population that really has not been able to participate in trials so far, and we're all eager to be able to do that. And I think there's an intention, if this looks promising in this population, to do a trial for juvenile Huntington's disease. The issue is, for this particular trial, is that the way juvenile Huntington's disease presents and progresses is different from people who get Huntington's disease as adults. And so to try to kind of keep and the data that we showed that we based the design of this trial on was in an adult population, people with adult onset Huntington's disease, from that PRIDE trial that I showed you. So that was the rationale for having that inclusion criteria. But I think if it does look promising, we would absolutely be interested, and I'm sure the study sponsor would be interested in doing a study that would include, in the future, doing a study that would include people with juvenile onset Huntington's disease.
A related question, a series of related questions. Maybe you could speak a little bit to how Prolinea is planning for testing this in other populations aside from early stage HD, for example, pre-symptomatic patients, or being able to administer to late stage patients.
Yeah, I know for a fact that Prolinea is interested in moving this earlier in the course of HD to the pre-manifest, prodromal and pre-manifest stages of HD, and have already started kind of thinking about what that might look like. So I know that there is an interest in that. There's no kind of firm plans about that trial at this point, but there are discussions about it. What was the other question? I'm sorry. What was the second?
There were some specific questions actually about whether it would be available in potentially in a liquid format. I was just sort of generalizing that to other stages of HD.
Oh, I mean, like more advanced Huntington's disease. Yeah, I mean, I think, again, as we've said, I think that if in this trial it looked promising and was working, absolutely, I think there would be an interest in either studying it in more advanced HD, or it's quite possible that if it looks promising and it's convincing that this works in this study, that the FDA would allow treatment of patients with more advanced Huntington's disease.
There's also a few questions about, some of the secondary endpoints and tests that you'll be running besides looking at TFC, and also as they relate to procedures. These 4 or 5 essential appointments that Sandra referred to. Will any of them involve a lumbar puncture or spinal tap to withdraw cerebral spinal fluid? And are there plans to look at, for example, mutant Huntington levels in the CSF of these patients? Because you had mentioned, I think on one of your earlier slides, that activation of the sigma-1 receptor could, among many things, it could activate and stimulate autophagy, which is the ability of your body to chop up and chew up maybe some of these mutant proteins. Is that going to be looked at in this study?
This is actually probably one of the easiest clinical trials from a patient perspective that's being promoted at this time. There are no PET scans, there are no MRIs, there are no lumbar punctures. There are blood tests and safety tests and other tests. We're looking at other outcome measures like cognitive function and motor function. But the answer, so because we are not doing lumbar punctures, we cannot look at a neurofilament or Huntington protein in the CSF. There's multiple reasons for that, but we want to get this proven effective, and that may address how it's working, but what's most important for us is to get an agent out that's working, and not necessarily whether those outcome measures, those other things like the amount of Huntington's in the CSF have not been proven to correlate with improving outcome. And what we're focusing on is the most important outcome measure here to prove that it's effective.
Yeah, actually, just going back to the previous question, I think the other question was about what other outcome measures are we looking at? And I'm sorry, I spaced out and missed that part of the question. We're going to be doing a number of other assessments, including the full Unified Huntington's Disease Rating Scale, which includes a motor examination, involves cognitive tests. We're doing a quantitative motor assessment that has been involved that we've done in many other clinical trials called Q-Motor. And we're going to be looking at patient-reported outcome measures, something called the Clinical Global Impression of Change, and others. So there's quite a long list of assessments that we will be doing in addition to the total functional capacity.
A lot of them are very similar to what if people have been in the Enroll-HD trial, a lot of the assessments are similar to some of those that people have already done in Enroll-HD, which is an observational trial where we're tracking people's progression over time with the CHDI Foundation.
Lots of people are asking about how to get involved in this trial and what it is like to get involved in the trial. Is there a screening at the beginning that would, a lot of people have specific questions about their own qualifications. Is that something that would be addressed if they went to this website or called this number, or would there be a visit involved?
We often do at our site, the research coordinators will often do a pre-screening call where people call in and say, what study do you have going? Or I heard about PROOF-HD, can I do that? And often they're actually patients from our local area. So sometimes we'll know them well, but sometimes they not necessarily. And they will actually often do a pre-screening on phone, because if there's something that's absolutely, contraindicated, it's not reasonable to have somebody come in for an in-person screening visit. So we usually do, and most sites usually do, a pre-screening phone call where patients can ask about the trial and what, the inclusion criteria. They can go over their medications with the coordinator. And then if it looks like they're a reasonable candidate and they understand once they hear the details of the trial, sometimes we'll actually send them out a consent form to review first, so that they can see some of the details of what their obligations for being in a trial are, and they can review those even before they come in, and they may decide, oh, this isn't for me, I don't think I want to do that. Or if they decide they do, we will set up a screening visit where they do come in and have screening assessments before enrollment. Those screening assessments will involve things like an EKG to make sure there's no cardiac problem that could be exacerbated, blood tests to make sure that their liver and their kidneys and their immune systems are functioning normally, and that they meet the other criteria of the study.
Yeah, and to get even to that stage, I think to find out if there's a site near you and which is the site closest to you, you could either go to this website, call the 800 number, or go to the HDSA's Trial Finder website as well. So yeah, I think that if you want to find out if there's a site near you or which is the closest site, once you've identified that, then exactly a phone call to the site usually would lead to a pre-screening assessment, and then if you check all the boxes, then to a screening visit.
There's just a lot of great questions coming in, and I don't know if we're going to be able to get to all of them because we are running out of time, but a couple quick ones I think that I'm seeing, and just I know they come up a lot, and just to reiterate, the the lower age limit is 25, but Andy, there is, to your knowledge, no upper age limit for this study, as long
That's
as
right.
you meet
There is
other
no
criteria.
No,
So
no
you
upper age.
can, thank you. So we are still seeing that question. So if you are in your 70s and you still meet the requirements, you could be eligible for this
Because it's
study.
very common for studies not to allow individuals above 65, but this study does allow individuals for above 65.
There's another question about the drug being potentially dangerous for pregnant women. I know that's an exclusionary criteria for this study. You cannot be pregnant and participate in this study. I don't know if there's anything else to add on that, but just wanted
Because
to throw that out
there's
there.
no way to know. So these kinds of trials, if you're planning on getting pregnant, you don't want to enroll in a trial like this. If you're already pregnant, you would not consider able to be in the trial.
And then one final one for me is which is also a good question, is do you require a family member or a caregiver to accompany the participant in these study visits?
Not required in the study. We wanted, as I mentioned, we mentioned that the total functional capacity range for the trial is 7 to 13. So we're expecting there will be people who have a 13 or fully functioning, with their Huntington's disease. And we wanted those people to be able to participate even if they didn't have somebody to accompany them. So it's not required.
However, if someone does want to have somebody come with, we
they're
kind
welcome.
of prefer that at least for the first and the last visits, when we're doing some of our assessments, that the same person comes and participates with the trial. But it's not a mandate that you have a caregiver for you with this trial. I also noticed as I watched some of the chats come in, there was a question, is there any fee to participate in the trial? And the answer to that is no. Study drug, placebo, study visits, there's no charge for these to participate in the trial.
Some folks are asking whether you take into account any kinds of measurements from other trials they may have participated in, for example, enroll or previous drug trials that they might have participated in.
I think that's a really good question. It's something that I think would make a lot of sense, but unfortunately we're in other words, if somebody's already had an assessment of whatever, do we need to repeat it again and that kind of thing? I mean, I think the answer is that we do, because we want to make sure that the investigator in this trial is doing the assessment and not a different investigator. But one thing we are planning to do is to look at the enrolled data for sites that are participating, and that way to try to identify subjects that might be eligible, actually. So that can be done, and we are planning to do that.
I had one question that was submitted via email, and since you talked a lot about receptors and the mechanism of action of this drug, I was hoping you could address it quickly before we wrap up. Basically, the question was, we hear a lot about dopamine in HD. How come we haven't heard about sigma-1, and what changed between the PRIDE trial and the PROOF trial that caused you to go in that direction?
Yeah, that's a good question. I think what happened, really, to be honest, was I don't know, because I don't know kind of internally what went on at the study sponsor during PRIDE and HEART and MERMAID. There was this idea that this drug was a dopamine modulator, actually, and so the thought was that it might affect motor function, in a positive way, even for motor functions that require more dopamine, and for motor functions that, like chorea, that can be improved by blocking dopamine. I think the modest effect of the drug led people to kind of question whether this was really the primary mechanism of action of the drug, and it led to additional studies. Using imaging of the sigma-1 receptor, and there's been a lot published about sigma-1 receptors and their role in neurodegenerative diseases and in Huntington's disease. So it's a natural thing to look at, actually, and it turned out to be highly selective for the sigma-1 receptor, actually.
It's not uncommon for investigators when they're doing research trials to learn something new about an agent that was designed, for instance, a medicine designed for managing seizures with GABA receptors. As the trial went on to identify other benefits of an agent where the molecule may not have been looked at initially as serving that purpose. And so there's several drugs which are on the market now, which have been, as we've gone through trials, discovered different mechanisms of action or better mechanisms of action, and drugs have been moved from one category to treating actually different symptoms. And that's sort of what's happened here is once discovering the effectiveness of the drug, looking at it in more detail since the beginning of the very first trial with Pridopidine.
Thank you. So we've had, literally dozens of questions, but we want to respect your time here. If folks have questions that they feel have not been answered, you're welcome to reach out to webinars@hdsa.org, and we will do our best to follow up with Dr. Kostyk and Dr. Feigin. George, do you want to wrap us up?
Yeah, no, I would just like to thank everyone for participating. The group, whoever was listening, this has been a really engaged group today, and we've had amazing questions. And I want to thank Dr. Feigin and Dr. Kostyk for taking time out of their schedule for presenting this to everyone. We are recording this, and I know we threw a lot at you, and we may have thrown some numbers and inclusionary and exclusionary criteria at you, but all of that is on the different websites, either on Trial Finder or clinicaltrials.gov or the Huntington Study Group.org website. So please do check out that, use that as a resource. But like Leora said, any other questions certainly do that we weren't able to get to, certainly reach out to webinars@hdsa.org, and we will try to address them as quickly as possible. So thank you, everybody, and we hope to see you all on February 18th when we do a recap of the great progress that was made in 2020.
Thank you so much. Thanks, everybody.
We appreciate the opportunity to talk about Pridopidine.
Thanks so much.
Thanks so much.
Thank you.
Take care.
Take
Bye.
care.
Bye.